cell cycle plays a key role while cell reproduction. And then this cell cycle is basically used while we are discovering new targets for drug discovery mechanism. Now this cell cycle basically is a key for pathways to discover new drug targets and ligands that are useful for making different drugs. This information is basically genome based, that this information is being used based on the genetic information of different organisms. Now what happens is that this cellular information or cell cycle, it is basically happening in different events or phases which are successive in nature, that one event occurs, then the other one, and then the third one, and so on. So what is the importance of this cell cycle phases? Phases are helpful while we say that if this cell cycle is regulated or not. Regulation is a step which helps us to find out if this cell cycle is normal or abnormal. So if it is normal, then it is producing new cells in normal conditions, or it is doing its functioning. On the other hand, if it is abnormal, then there are chances that if there is cancer present, or there are some errors or malfunctioning of cell cycle. Moving on, how does this cell cycle is regulated? There are different checkpoints. These checkpoints help us to repair the damaged cells so that cell keeps on working in the proper format. So when we talk about phases, there are 4 major phases of cell cycle. If I start those phases, the first phase is this G0, which tells us that this is the non-dividing cells, that these cells will not be divided and will not produce other cells. Then when we move towards other phases, first phase, which is this G1 phase, which is shown by this part, complete part, what happens in this one is that cells increase in size and synthesize RNA plus protein, which is written here also, that there are cellular contents which are related to a given cell, excluding the chromosomes, and these cellular contents are duplicated, which is shown here also, that we have cellular components, and from this first cell they are duplicated. In the S phase, which is basically a synthesis phase, what happens in synthesis phase is that we have DNA replication occurring in this phase, that DNA replicates. And so what happens, for example, for a human being, each of the 46 chromosomes in human beings are duplicated by the cell in this phase, as shown by here. Then we move toward G2 phase. G2 phase, what happens in G2 phase? We have cell growth and new proteins produced. So as shown in this figure, we have this phase, basically cell double checks the duplicated chromosomes for error and making any needed repairs. So what happens is, after G2 we have this thing, which is the new cell. After that we have this mitosis phase, which is shown in this part. So this is basically a part where this cell division is being occurring. So if I divide this complete figure in two things, this is mitosis part, which is basically when the cell divides into constituent parts, and these are other remaining things which are shown. We can say that they are interphase where the cell works in its normal condition. So there are two parts, interphase and mitosis. Then what happens is, this cell cycle phases, they are basically working with the help of time. So if I divide these phases with the help of time, what I will get, that this G0 phase, first phase, basically it takes 9 hours. Then from G1 to S phase we take 11 hours, and similarly from S to G2 we have 3 hours, and finally 1 hour for G2M phase. So these are different information related to these phases with respect to temporal information. Similarly, as we said, that mitosis is the phase where the cell divides into its different parts, which is shown by this figure. This mitosis, it can be further divided into 4 different phases, which are prophase. Now what happens in prophase, that chromosomes become visible and the nucleus disappears. So we only have the information of chromosomes. Then what happens in the next phase, which is a metaphase. In metaphase, what happens, we have spindle fibers which line the chromosomes along middle of cell nucleus. So its division is such that these fibers, spindle fibers जो होती हैं, वो chromosomes के middle में आ जाती हैं. So the chromosomes can be seen separately. In the third part, this anaphase, the chromosomes which were in the different phase or which were in the middle of the spindle fiber, they get separated, वो separate हो जाते हैं, and then they move to the opposite direction to each other. And finally in this telophase what happens, we separate cell generation कि different या separate cells produce हो जाते हैं in the telophase. So if I summarize these 4 steps, there are 4 steps. Number one is G1 phase, in which cell increase in size, produces RNA and synthesizes proteins. Then we have S phase, which is also called synthetic phase, in which DNA and centrosomes are generated or synthesized. Then we have G2 phase, in which cell grows and produce new proteins that are needed for mitosis step. And finally we have mitosis stage or M phase, in which we have cell growth and protein production is stopped. Each cell undergoes a complex mechanism and then divides into its daughter cells. So for details of this information, I have taken an example which shows more details of these 4 phases, which are basically related to mitosis. So what happens, this is our initial cell basically, which has nucleolus, chromosomes and nuclear membrane. During interphase what will happen, cell grows and develops, के cell बढ़ता है और अपने आप को और produce करता है, और इसके end में after the interphase DNA double हो जाता है and chromosomes also become double, so which is shown here. So what will happen during the prophase phase, chromosomes become visible as shown here. There are 4 different chromosomes. Then nuclear membrane disappears, so nuclear membrane खत्म हो जाती है और nucleolus भी disappear हो जाता है. In this metaphase, these are our spindle fibers जो हमारे middle के अंदर appear हो जाती हैं as shown by this figure. Then what happen in this anaphase stage, that chromosome split, ये 2 parts में separate हो गए हैं 4 के, और each half is pulled to opposite side of the cell कि अब different side पे cells आ गए हैं. During the telophase what happens, chromosomes cluster in the center of each new cell. अब यहाँ पे आप देखें ये cell हमारा बनने की तरफ हो चुका है, and then nuclear membrane begins to form और cell begins to separate. And finally in cytokinesis what happens, cell बन चुका होता है और finally उसका cytoplasm भी divide हो जाता है और 2 daughter cells बन जाती हैं और they have the same number of chromosomes at the parent cell. And finally we have this single cell out of this information. So starting from simple cell, it goes on dividing and then finally a new daughter cell is produced from the parent cell.